An individual's genetics could influence their susceptibility to particular drug adverse effects, and we really want to understand how this process works, how it's triggered, and then also how we can use that understanding to better harness therapeutics to improve health of cells and tissues within the body.
I think there's a huge therapeutic endeavour to actually personalise medicine. We can even see this already happening with the personalised mRNA cancer vaccine programmes, where mRNA vaccines targeting an individual's specific tumour are already being developed. But the side effects are extreme, and my group focuses on both why immune responses are different in people and on making safer mRNA therapeutics.
Individuals respond differently to different immunotherapies due to a range of different factors. So some people might respond differently due to their age or environmental factors, and others might respond differently due to more inherent factors such as inborn errors of immunity, which mean different parts of their immune system are not functioning properly.
But even things that we don't currently consider, things that impact the immune system, like your body weight. One of the things we found, for instance, is mRNA vaccines didn't seem to work as well in people with obesity. So understanding what impact weight has on immune responses is a huge problem for society.
That's something we're investigating in the lab right now in a variety of different models. I'm looking at what molecules are signalling in the immune response in these individuals and how we might correct these signalling pathways in order to improve the response to vaccination in these individuals.
We've also found that mRNA vaccines used in the COVID pandemic can actually trigger off-target immune responses.
So immune responses to parts of the vaccine that shouldn't be being made. So the off-target effects we see in the case of the COVID vaccines aren't actually harmful themselves, but when we have those immune responses being generated, we're actually getting an overall reduced immune response to the thing we're trying to get a good immune response to. We use a combination of cell models and animal models to look at these off-target responses, and this information is then helping us learn about different ways we can modify these RNA vaccines to help reduce these off-target effects. So when we've actually done that in the lab, we can see that we stop these off-target effects, and then as a consequence of that, we get enhanced vaccine response overall.
So it's practical to actually start giving personalised therapies to people based on their genetics, their body weight perhaps. So there's no reason why we can't actually target the individual characteristics of people to make better and safer therapies.
My group here at the MRC Toxicology Unit focuses on mechanisms of cell death or apoptosis. Apoptosis is essentially a process that regulates cell numbers in the body. Apoptosis is important to understand in the context of therapeutics because certain therapeutics, for example immunotherapies, they may have an impact on pathways that are regulated by apoptosis. We're exploring whether or not there could be a genetic underpinning to that by sequencing mitochondrial DNA, because there could be individuals that have a mutation in their mitochondrial DNA which makes their mitochondria more susceptible to the adverse effects of certain drugs. So in addition to the work that we do in wet lab work here at the bench, we also have an approach where we're trying to involve machine learning models. To build these models, we feed in our wet lab data, and then we can use that trained model to ask whether or not some unknown drugs or chemical compounds that are perhaps already in use or are under development, whether or not they would also induce mitochondrial toxicity. The application of machine learning models really doubles up our effort because we have our wet lab work, but also we can screen, using this in silico approach and machine learning model, a much larger number of compounds in a shorter time, so it's much more efficient.
I'm primarily a clinical researcher. I look after the problems of real patients that we see in the hospital, and it's brilliant being embedded in a unit surrounded by basic scientists, as well as cutting-edge technological developments and scientific developments that are coming from the other groups, and see how we can work together to practically turn that into improved healthcare.